Okay, good morning. Let's continue our discussion about how cells respond to different types of signals that drive the function of certain proto-oncogenes forward. So let's look at things like growth factors today. We'll take a closer look at SRC and how SRC actually functions, or its purpose in the cell, and also look at a very important group of proteins, especially for cancer, known as the receptor tyrosine kinases, or RTKs. So let's start off with a quick sort of check-in here of where we're at in the semester. So now we are starting week 4. So here's our Monday lecture. Reminder, this Friday is your second article discussion. So that means Wednesday of this week, you'll have 2 things due, right? You have your quiz that'll be due in the morning, and that quiz is on chapters 4 and 5. So after today's lecture, which is the chapter 5 stuff, I'll make that quiz available, and then you'll have until Wednesday, 10:30 in the morning to take it. But the more sort of involved thing is you have your article summary that's due Wednesday night at 11:55. Right. So as we continue through the semester and we talk about more and more topics, you should feel free to choose papers that have to do with any of the topics that we've discussed so far that you find interesting. Okay. So just a reminder that the article summary assignment is available now in La Lima. So you can go ahead and submit it at any time. Right. Then next week, we will have Monday's lecture will be the last new material before the exam. Then on Wednesday, we will meet as normal for a class, but we'll just do a review session. I kind of look forward to that because then, I'm not going to lecture, but we can just all talk and you guys can ask questions and we can talk about things that were maybe a little bit ambiguous. And then on Friday will be the exam. And so the exam is a combination of multiple choice and free response. And you'll just take it during normal class time. So everybody logs on to Zoom. So that myself and Zoe and Alexis, the TIs, we can help proctor it. You'll have your cameras on. So one thing I'd like you to do is make sure that you have a working webcam. If you don't have a working webcam, like the one on your laptop is busted, then you can do the thing where you have your cell phone maybe logged on to Zoom so that we can see you. And then all of you would take the exam over La Lima. All right, so it'll be available in La Lima. All right, so that's where we are. Any questions about any of that more administrative kind of stuff? Let me activate my chat box because I realized I can have the chat open at the same time. Well, I don't know, it's not showing up now. All right. So here's some thought questions to kind of start off our lecture. So proto-oncogenes are often key molecules in cytoplasmic signaling pathways, right, that interpret extracellular stimuli. We looked at examples such as RAS or SRC as being sort of like these supervisor kind of proteins, right? If you were to have different maps of signaling pathways involving many proteins in the cell that maybe start from, let's say, the membrane and end up eventually activating different transcription factors to control expression of different genes, we said that these proto-oncogenes like SRC and RAS are sort of these, mid-level supervisor ones, right? But we learned last time that they have to respond to like the big boss. And in the case of the membrane, we're talking about things like growth factor receptors, like epidermal growth factor. And that growth factor is going to respond to different messages that are arriving on the outside of the cell, activate, and somehow then give instructions to proto-oncogenes like RAS and SRC so that they start signaling. So if you think about that picture then, what kind of insight does that give you about how normal cells decide what to do at any given time, right? So a cell, it's constantly capable of all of these things. It's capable of differentiating, right, to become like the full form of the cell that it's meant to be. Cells are able to proliferate, right? All the information necessary to proliferate and divide is in there, or even things that are even more fundamental, like how does a cell know that it should stay functioning, right? Survival versus things like apoptosis, right? Where it ends up dying. How does a cell even know to stay put versus move around? Right? So now that we have this picture forming, I hope what you begin to realize is that a cell in isolation by itself doesn't really make these decisions on its own. Right? These decisions that have influence or encourage different phenotypic outcomes has to do with the cell being told what to do. Right? And oftentimes, it means these individual cells normally do not make decisions about these events on their own. They require some kind of feedback from other cells. And then these cells will do what they're told to do if they are a normal, well-behaving cell. So let's explore this a little bit more. If we look at something like a complicated tissue structure in our body, so this is, I think this is your small intestine. So the lining of your small intestine has these different villi on them. So the function of the villi, which are made of thousands of cells, is to increase the surface area, for example, of your GI tract lining to maximize absorption of nutrients. It's also to do things like, protect the tissue underneath, for example. Right? So in this typical cross-section structure here, you can see that the outer layer of the villi are made of epithelial cells, right? So the epithelial lining would be the outermost lining that goes all the way around. And those epithelial cells are a mix of different types of epithelial cells. Some of them are glandular. So these pink ones are the mucus secreting goblet cells, right? So remember, depending upon what type of cell in this epithelial lining that starts going bad or going rogue and transforming, you might end up with something like a glandular cell leading to a tumor, and that would be our very classic adenocarcinoma, right? Glandular cell that ends up transforming. We will explore later some reasons why things like adenocarcinomas are very common compared to, let's say, any of these other epithelial cells around it, being the ones that turn into a tumor, right? So there's some very good reasons why that's the case. So underneath these would be your, it's kind of tough to see, but there'd be a basement membrane layer of proteins, and then you'd have a core of mesenchymal cells, right? So these smaller purple ones going through the middle, these are your mesenchymal cells, or we also call it the stroma. So this is the supportive connective tissue that is helping to communicate and helping to organize the epithelial cells that are on the outside here. So how do these epithelial cells of the intestine work together? So there's got to be a lot of cell-to-cell communication. For example, right, these epithelial cells need to know when to multiply so that they can constantly be replenished. Keep in mind that because this is the outermost layer of your tissue here, so all of the sort of pale greenish white space at the top here, this is the lumen of your intestine, right? This is the inner space of your intestine. So the food, your food that you're digesting is all pushing through here, right? And actually rubbing up against these villi. So a lot of these epithelial cells just get shed, right? They just get sloughed off because of the physical force of the food moving over them. So clearly your epithelial cells, right, or the cells that differentiate into epithelial cells, right, they need to constantly know when to multiply to replenish that, right? And how would the epithelial cells know what spot to occupy on here, right? So if there were an area of epithelial cells missing, how would the other epithelial cells know to go and fill it in? So it all has to do with these cell-to-cell communications. So here's an example. Here we have some fibroblasts. So these are fibroblast cells. So these are cells that often make a lot of the tissue that's in different types of epithelial layers. And these cells are a good example of how cells respond to different types of growth factor signals. This is a very classic type of experiment that's often done in research groups who study things like wound healing, like when you get a cut, right? When you get a cut and you actually end up having many of your epithelial cells damaged or absent at that site because of the wound, how do the surrounding epithelial cells know to kind of stop just doing what they're normally doing and instead focus on going into the wounded area? So in these experiments, they grow a monolayer of fibroblasts here. So here they have a layer of fibroblasts on the dish, and what they've done is literally taken like a little scalpel or needle and scraped the surface, right? So they've scraped their way through these fibroblasts. Right? So in a control situation here, where the fibroblasts are just kind of left to grow there, right? Nothing much happens because this is very artificial. This is in a plastic dish. And so the researchers have control over what's in the media, what types of things are there, right? It's a very unnatural situation because these epithelial cells, right, no longer are part of the tissue, normal tissue environment that includes other cells that communicate with them, right? They're just in isolation by themselves. So if you leave them alone, nothing much happens. Okay? But if you give these cells some platelet-derived growth factor, right, PDGF, and you mix that in the media, what you see is after 48 hours, those cells have now migrated to fill in the wound, right? In this case, the scratch on the plastic. Right? So that's what happens if the cells are wild-type normal cells that have the correct receptor on their surface for platelet-derived growth factor. So these cells, in other words, are PDGFR, that's the receptor, positive, right? They have the receptor on their surface, so therefore they can respond to the PDGF that got mixed in the media, right? So the PDGF in the media ends up attaching to the receptors on the surface of these cells, and then what you see now is the eventual result of those receptors telling the cells to change their phenotype and do things like start migrating, right, or proliferating. But what if you have cells that are mutants that lack the receptor? You would imagine then that in the case of the controls, right, it probably should look the same, but what would it look like if you then added PDGF in the media, right? It looks like this, right? Much reduced evidence of the cells responding to the platelet-derived growth factor, right? Because they don't have the receptor on their surface in order to pick up on it. So so I should point out, right, that where do those PDGF signals normally come from? Well, they come from other cells in your tissue, right? So if your tissue gets damaged and the epithelial cells get damaged or missing, right, the other cells in the area will start doing things like making PDGF. By the name, you can tell that PDGF is often part of platelets, right? So platelets are fragments of larger megakaryocytes, right? So platelet-derived growth factor are growth factors that are released by the platelets that are going to the site of the wound, right, to help with the healing. So not only do the platelets help with clotting and healing, but they also start stimulating the growth of epithelium. So if we look at a cell then, so this gray part is the cytoplasm of the cell, and then inside, the brown boundary here is the nucleus where the genome is. What we've been looking at so far, primarily the last few lectures, was beginning to understand the function of certain proto-oncogenes like RAS, right? We said that RAS flips between its off and on state by binding to GTP, right? And then RAS will pretty quickly catalyze the cleavage of GTP back to GDP. So it ends up turning itself off, right? And then RAS will signal many numerous downstream proteins to eventually get transcription factors active on different genes. Right? So now what we're talking about is what's above this. Right? So what's above this then is a whole array of different receptors that are important for controlling things like RAS, right, and activating it. And importantly, these receptors normally, right, if they're behaving well, are the right shape to wait for the appropriate external signal to arrive, right, such as epidermal growth factor, right, epidermal growth factor that will arrive and bind certain of these receptors, activating certain pathways that then turn on eventually different genes that control things like apoptosis, cell migration, growth, adhesion, and differentiation. So today what I'd like to do is take a look at some of these up here. First off, I wanted to go in a little bit more depth as to how Sarc works. All right. So Sarc works as a kinase. So kinase is a protein that takes a phosphate group and puts it or transfers a phosphate group onto a different target. And that other target might be, like another protein, for example, thereby phosphorylating that protein. And by binding a phosphate group to that protein, it might change that protein's shape, which activates it and causes it to engage in some sort of downstream activity. So this illustration in the book, it's kind of funny. This is meant to be Sarc, but it's kind of a funny figure, right? Because clearly what it's trying to show is there are important little features. There's a catalytic cleft in here, kind of looks like a mouth or something like that, right? So I remember the first time I saw this in the book, I'm like, this reminds me of something, right? I couldn't tell what. It looks like some sort of cartoon character. And then for the longest time, I couldn't figure it out. And then I realized, oh, it kind of looks like Shrek, right? So anyhow, all right. Well, so I don't want to mention that because then you're just going to be all distracted staring at this and trying to imagine Shrek being there. But what I really want you to do is pay attention to what's going on over here. All right. So it turns out that the way Sarc works is that this catalytic site is able to convert ATP, right, to ADP, and in the process, it takes the phosphate group from it, right? That phosphate group then will be transferred to another target. The reason why this is such an important sort of example is that when researchers were first trying to study Sarc and figure out, what does Sarc actually do that makes it so important in the cell, especially when it has to do with cancer, is, at first they didn't realize that it's a kinase, but they wanted to try to study it by labeling Sarc protein, right, by using antibodies. So they designed antibodies that the antigen binding domains of those antibodies are specific to binding Sarc. Right? And they thought, great, now we have a tool for being able to, let's say, highlight or stain for Sarc protein in different cells to make it easier for us to see Sarc in different tissues. But an interesting thing happened. They discovered, strangely enough, that when the Sarc antibodies labeled the Sarc protein, the antibodies ended up gaining a phosphate group, right? So you can see where this is going, but it's kind of a cool accidental way of discovering that what Sarc does is it transfers phosphate groups onto target proteins that bind to it. So that's how they first learned that Sarc is a kinase. So that's way back in 1977. I was just a little kid back then. All right. So researchers found that the antibodies that were designed to detect the Sarc protein would often become phosphorylated. And the important thing here is not just on any amino acid residue of the antibody protein, right? Because remember, antibodies are proteins, right? But specifically on tyrosine residues on the antibody, right? And you're like, so what, right? So Sarc specifically phosphorylates tyrosine residues on the targets of its proteins that it interacts with. So why is this important? So Sarc is a tyrosine kinase. It's important because this process of phosphorylating proteins to activate them, it's a very common mechanism that your cells use to activate proteins, right? I think you realize that. But maybe what you don't realize is that more than 99% of the phosphorylation events, proteins being phosphorylated in your cells, involve those proteins getting the phosphate group attached to a threonine or a serine, right? So more than 99% of kinase events or phosphorylation events that take place to influence the function of proteins take place by adding phosphates to make phosphothreonines or phosphoserines. Right? Only around 0.05 to 0.1% of all the phosphorylation events, protein phosphorylation events that are happening inside of your cells actually involve the creation of phosphotyrosines. Right? So here is a tyrosine residue. Right. So in this particular chain of amino acids, the previous one's just like an R group somewhere. So whatever that amino acid is, and the one downstream of this is also some other amino acid. But the one in the middle here is a tyrosine, right? And it can get phosphorylated in very, very rare situations. Why is this significant? Think about it this way. The majority of phosphorylation events that involve threonines or serines, this is kind of like, let's say you wanted to go to a big concert, right? So like a couple of years ago, the hottest ticket in town was like when Bruno Mars was playing Aloha Stadium, right? So yeah, if you're lucky, you can be one of the tens of thousands who managed to get some tickets, right? But these are just normal tickets. This gets you into Aloha Stadium, gets you to your seat, right? And then you have fun at the concert, and then you try to get out of the parking lot later, right? But let's say instead you had this VIP all access pass, right? That means after the show, you get to go backstage and meet Bruno Mars and hang out, right? So clearly, in the case here, right, very specific privileges, right, are given to you depending upon what type of ticket you have. And it's a way to kind of separate out, right, sort of the general population, right, from the subset of individuals, right, who deserve special treatment, or to put it this way, right, are kind of monitored even more closely and get certain access. So that means tyrosine phosphorylation, when it happens, evolutionarily is reserved for very critical special functions inside of the cell so that they don't get kind of confused with the normal threonine and serine phosphorylation, right? So think about it. What types of signaling pathways evolutionarily, right, deserve very special care, very special attention, right, and very special monitoring to make sure that they are carefully controlled? Right? Well, things like pathways that, if they are not properly controlled and specially set aside, can lead to cancer, right? So proliferation, cell cycle, apoptosis, differentiation. Right? So these special tyrosine kinase pathways are very important in cancer. So a number of those big boss receptor molecules then on the surface of the cell that are crucial for driving these potentially cancerous pathways, not surprisingly, they are not just kinases, but they are tyrosine kinases. Right? And because they are in the form of receptor proteins, we call them receptor tyrosine kinases, or RTKs. So here is a selection of different examples. All right, so many of these have these different cysteine-rich domains on the outside. Some of them have like these more immunoglobulin-like domains that are closer related to certain types of immune system proteins. So they vary quite a bit on the outside. But once you get through the membrane, so of course they all are anchored to the plasma membrane here, this is the gray. In the cytoplasm, the cytoplasmic domains that stick into the cell, where the actual communication with cellular proteins take place, they tend to be a little bit more homogeneous, right? And importantly, they all have this sort of red rectangular feature here, which is the tyrosine kinase domain, right? So this is the domain that handles managing the phosphate group and transferring it onto targets. So you can see here's epidermal growth factor, here's insulin-like receptor, right? Here is the PDGF receptor, right? So it also has these tyrosine kinase domains. And what I wanted to point out is that if we were to look at an individual one of these red rectangles and look at its amino acid sequence, what would you find? Oh, right, they have very high homology with the SRC tyrosine kinase domain, right? These are those SRC homology type of domains, right? Or the SH domains that are conserved through evolution. Remember, they're called SRC homology domains because they were first identified in SRC, right? So they are the mouth of, who is that? Shrek, right? And so that amino acid sequence that makes that very important catalytic domain is used over and over again by various other proteins that also engage in this highly controlled tyrosine phosphorylation type of mechanism. So let's take a closer look at the activation of growth factor receptor tyrosine kinases, right? I'm just going to call them RTKs now that you know what they are. So in a normal cell, you would expect to find these RTKs, if you chose one particular type of RTK and looked for it on the surface of a cell, you'd find that they're present in relatively small numbers, right? Because again, the fewer of these you have, right, the more control you have over making sure that they are activating properly. They are found as single inactive monomers. You know, we already looked at this in a brief example a couple lectures ago when we looked at things like epidermal growth factor receptor, right? We said normally you would have two of these monomers just kind of floating around on the surface of the cell. Keep in mind, right, that even though they're anchored to the surface, right, that just means because of their hydrophobic residues in the middle, they won't float out of the cell and they won't float into the cell, right? The hydrophobic residues keep them in the hydrophobic layer of the cell membrane, but they are free to kind of drift around on the surface. I oftentimes say, think about proteins on the surface of a cell membrane as being like a bathtub that has a whole bunch of little floaty bath toys in it, like rubber ducky, tugboat, whatever, right? They are free to float around, but rubber ducky and tugboat aren't just going to magically float up and away, nor will they sink to the bottom, right? Unless you shove them down there. They're always on the surface, but they can interact with each other if they bump into each other by chance, right? So these individual monomers of RTKs, there's relatively few of them so that you actually discourage that chance of them coming together by themselves, right? They're just kind of floating around. Yeah, I even say they freely wander around. So what happens then? When something like a growth factor arrives, right, it encourages the two monomers to come together. Now they're kind of buckled together because their tops each hang on to part of this growth factor, and that brings the two monomers together to form a dimer now, right? And once the dimer is formed, that combination of the two monomers together, that is going to activate signaling inside of the cell. So this would be a normal healthy cell, right? Very few of these, right? They're the right shape to wait for growth factor, and then growth factor causes dimer formation. Dimer formation causes their cytoplasmic tails to work together to then activate. What about cancer cells? So we already looked at it with the example of the EGFR or epidermal growth factor. What if you had a mutation in the gene that codes for this particular receptor so that you now have a stop codon, right? A premature truncation of the protein that has removed the ligand binding domain. So it might be that these truncated receptors now, right, might come together a lot easier because they're not inhibited by the ligand binding domains not being the right shape to kind of come together, right? So that might improperly encourage the receptor to start firing off, even though the cell has not received the message in the form of growth factors. Or another mutation, you know, we tend to think of mutation as being these mutations that actually change the coding sequence. But one of the things that I've been trying to teach you guys is that mutations can also lead to overexpression or underexpression of a gene, right? If the mutation actually happens in, let's say, a regulatory region, or it has to do with translocations, right? So a certain gene gets put somewhere else in the genome where it is now under the control of a highly lenient promoter that's constantly now expressing it. So if you get too many of the individual monomers floating around, then the chances of them bumping together, right, is increased. And so if they bump together, then they might signal. Okay. So normally, how do cells communicate with each other then to kind of control these very important messages? One of the important cell-to-cell communications that we discuss in this class is the communication between the epithelial cells, right, and the mesenchymal cells beneath them that talk back and forth so that the epithelial cells know what they should be doing. Right? So on the left here, we have an epithelial cell. And on the epithelial cell, it expresses this epidermal growth factor receptor. Right? On the mesenchymal cell, so on the other side of the basement membrane, the mesenchymal cell doesn't have EGFR. It has platelet-derived growth factor on it, so the orange one. When the epithelial cell makes products like the platelet-derived growth factor, right, the epithelial cell itself doesn't respond to the PDGF, right, because it is not expressing the PDGF receptor on its surface. But as the PDGF is being made, right, and it kind of like forms a cloud of PDGF that's kind of coming out of this epithelial cell, right, some of it gets through the basement membrane and it communicates with the mesenchymal cell. Right, so in this case, let's say the epithelium got damaged. And so as a part of signaling damage, right, some of these damaged cells, right, are making PDGF, and the PDGF arrives at the mesenchymal cells. And in response to the PDGFR receptor, the orange receptor being activated, right, the mesenchymal cell starts expressing certain genes, for example, TGF alpha. Right? And TGF alpha is released by the mesenchymal cell, and a cloud of it is kind of emanating from the mesenchymal cell. That is the epidermal growth factor that, let's say, this particular EGFR is designed to bind, right? And then that stimulates the epithelial cell, and these epithelial cells get the message then, oh, what we're being told to do is go ahead and divide and start moving around to try to fill in, let's say, the actual wound, right? But keep in mind, the epithelial cell would not know to do that, right, unless it received the instructions from the supporting cells underneath. Right? So there's this back and forth communication. But what if certain mutations happened? Right? Let's say in a normal cell, the epithelial cell, is not going to make the growth factor that it has receptors for. Right? So in a normal cell, those growth factor receptors are just sort of floating around waiting to hear from a different cell to send it messages. But what if you have a mutation that then activates the expression of that same growth factor that the cell has a receptor for? Right? So another good example of how mutations can work, right? Not just affecting the coding sequence of a gene, not only affecting, let's say, expression like higher or lower, right? It could very well be that a mutation actually turns on a gene that normally is shut down in the cell. In this case, this epithelial cell then makes the very growth factor that it has the receptor for. And so then as a result, this mutation causes this epithelial cell, right, to start acting independently of messages from other cells. It starts doing its own thing, and you have this autocrine signaling loop. Okay. How do these receptor tyrosine kinases start signaling? Now, we already know that it has to do with if you have two of these monomers floating around and they're brought together, normally because they both attach to an epidermal growth factor molecule, some kind of a ligand. And we already said that causes the two cytoplasmic tails to come together. And then in previous drawings, then there's like some like rays of light or something, meaning that it's activating, right? So let's take a look at what's actually happening. When these two tails are brought together, remember these tails are made of these tyrosine kinase domains, right? That if they are brought close to another protein will have a tendency to facilitate the phosphorylation of that target protein. So in this case, the two monomers, right, they are the other protein that's brought close up against it. And so the two tyrosine phosphorylation domains begin to phosphorylate the other monomer. Okay, so the one on the left begins to phosphorylate, put phosphates on tyrosine residues, right, that are found in the tail of the monomer on the right. And the monomer on the right, its kinase domain starts putting phosphate groups on the tyrosines of the one on the left. Okay. And that event then causes the tails to kind of change shape, right? Because these polypeptide chains, they begin to change shape and they start assuming certain shapes that other proteins that otherwise would just be kind of floating by and bumping off of them now are the right shape for those proteins to associate with them. And that's how you kick off a chain of events inside the cell. Okay, so we will talk more about RTKs, in particular, different examples as we start looking at particular pathways. But I wanted to point out to you that there are many non-RTK type receptors that are also involved in cancer. Right? So these are receptors that may also involve phosphorylating things, but not necessarily tyrosine residues, or these are some types of receptors that aren't anchored to the surface of the membrane. How we typically think of a receptor is it's anchored there, right? But in reality, it doesn't have to be anchored there in order to wait for something like a ligand to bind it and then send out messages. So some examples that we'll look at today are things like cytokine receptors that work by recruiting kinases to them when they're activated. In other words, coordinating the actions of other kinase molecules. We have ones where you have one receptor molecule acting or activating a different type of receptor molecule. We have receptors that are actually traveling within the cell itself that are not bound to the membrane. And we have ones that are bound to the membrane, but don't necessarily wait for free-floating messages in the form of like growth factors or whatever. There are certain receptors that are interacting with just the environment around the cell to be able to tell the cell something about the surrounding matrix. Right? So let's quickly look at these, and that will kind of wrap up our discussion for today. First one are cytokine receptors. So cytokines are small protein signals that typically come from immune cells. So when your immune system activates many of the components, cellular components like white blood cells, they need to talk to each other, or certain cells of your body, if they are infected with something like virus, they send out different cytokine messages to alert your body to infection. So things like interferons that are sent out as little cytokine messages from cells that are infected with the virus, or interleukins, which are the little protein molecules that are sent between different types of leukocytes or white blood cells in order to coordinate the function of white blood cells in your immune system. Those are all cytokines. So typical cytokine targets are cells that are typically involved in your immune system. So erythroblasts, so the cells that give rise to red blood cells, right? They respond to different types of cytokines. Megakaryocytes, which are the humongous cellular precursors of platelets, right? They receive cytokine messages so that they begin to break apart and form little platelets. And of course, white blood cells, as I mentioned above. These cytokine receptors recruit kinases to them in order to carry out their function. So an important group of these kinases are the Janus kinases, or the JAKs. So the very first Janus kinase that was identified was just called JAK1. Right? So Janus is, I think, the Roman god of beginning and end, right? So I think Janus is often portrayed as being two-faced, one looking in the past, one looking in the future. And that's the reason why it was named that way. Hopefully will become evident as I explain how they function. So here you have two monomers of a particular cytokine receptor. These are alpha interferon receptors, right, that are going to receive a cytokine message, alpha interferon, that might come from virally infected cells in the tissues of your body. These alpha interferon receptors, they recruit different kinases to them. For example, this one's recruiting JAK1. This one is recruiting another Janus kinase called TYK2. Right? Normally, right, the JAK1 and the TYK2, they just be floating around somewhere, and it's pretty rare that they would bump into each other by themselves. Right? But because they are associated with these interferon receptor molecules, once the interferon arrives and reorganizes or arranges the two halves of this cytokine receptor to come together, then the two Janus kinases are very close to each other and they can act on each other. Right. And so one of the first things that happens is they activate and they phosphorylate each other. Right. After they phosphorylate each other, they then phosphorylate the tail of the receptor. Okay. And so this is why it's called Janus, because there's a beginning part of it, which is the phosphorylation of each other, and then the end part of it, which is the phosphorylation of the tail of the receptor that then starts talking to other proteins. Right? I wanted to point out this term here, right? Because this is the blue TYK2 molecule phosphorylating the green JAK1 and also vice versa. This is two separate molecules phosphorylating each other. So we call this transphosphorylation, right? Just like the two individual monomer tails of a receptor tyrosine kinase that we looked at in the previous one, when they are phosphorylating each other, that can be considered transphosphorylation as well. But the terminology gets a little tricky, right? Because when we were talking about the receptor tyrosine kinase, right, that doesn't have other kinases actually doing its thing, it itself is two kinases that are phosphorylating the other strand. That is trans, but because it's the same activated receptor, that's also considered autophosphorylation, right? It's the same complete receptor phosphorylating itself, although technically there's two halves that make up the receptor, and the two halves are engaged in transphosphorylation. I point that out because oftentimes, even in the literature, right, if you were to look up an article about this, they tend to be a little sloppy, referring to it as transphosphorylation or autophosphorylation. But hopefully, if you think it through, you can see that the terms can actually be, one and the same, depending upon the situation. All right. Here's another example. This is transforming growth factor B, or TGF-beta receptor. So in this case, it is not even a tyrosine kinase. It is a typical serine-threonine kinase domain. In this case, the binding of TGF-beta allows two types of the TGF receptor to come together, right? A type 1 and a type 2 are brought together, and then the type 2 phosphorylates the type 1. And then the type 1 then changes its shape. And that's the surface, which other proteins then can bind to. A special type class of receptors are known as nuclear receptors. So these are receptors that are not fixed. These are not anchored to the surface of the membrane. They're just floating around inside of the cell. Right? So think of a receptor as not necessarily having to be anchored to some membrane surface. I mean, I know classically, that's the easiest way of picturing it, because that's how we can picture things from the outside arriving on a cell, and then the receptor, because it changes shape, is able to transduce that signal into the cell, right? Even though the actual molecule that arrived on the outside never goes in, right? That's sort of the classic receptor, how we picture it. But if you just look at any molecule that has the ability to have an input and an output, I mean, that is a receptor. Right? The fact that it's not actually anchored somewhere doesn't mean it's any less a receptor. So in this case, you have certain proteins that float around. And if small ligands attach to them as the input, right, then that causes the receptor to do something else that ultimately has an output, meaning talking to other proteins downstream. So a good example are things like hormone receptors. Here we have a hormone. So maybe it's a small steroid, for example. Maybe it's testosterone. Maybe it's like estrogen, right? Or it could be other steroids like glucocorticoids that are involved in inflammation. They're carried around in your blood with some protein carrying them around, so this way they don't get eliminated in your urine. Some of them will make their way eventually, by chance, into the tissues where the cell type that they are meant to talk to is found. So here's a cell, right? So you would have certain receptors on the surface of the cell that might be, receptors that receive them that way. But what I'm talking about here are these cytoplasmic nuclear receptors, right? They also receive the yellow dot. And when they bind the yellow dot, these go into the nucleus. That's why they're called nuclear receptors. And they act as transcription factors. They attach directly to things like promoters or enhancers of the genes that they're meant to control. And they help with the assembly of other transcription factors that will then result in the expression or control over the expression of a particular gene, right? It might be the recruitment of RNA Pol II to turn on expression, but it also might be a reverse effect, right? It might be that by binding there, the nuclear receptor interferes with the RNA Pol II, and that would shut down the expression of a particular gene. But the protein product of that gene then, of course, gets translated, and you get some new product that's being made, okay? And the last example are these integrins. So integrins are also these surface-bound receptors, but they are not, kinases or something like that. Their purpose is to help detect the nature of the surrounding around the cell. There are a number of different types of integrin molecules, and again, they work as a dimer. But in this case, right, it's like a mix and match. Depending upon which pairs of monomers come together, these integrins have different function. You can see on the left, this is a very classic example where one is this green kind of alpha subunit, and the other one is a blue beta subunit, right? And they have very peculiar shapes in their extracellular domain. And so depending upon how they're brought together, you get a combined sort of shape here to bind things. So you can see we have a number of different alpha subunits to choose from, a number of different beta subunits to choose from, and depending on how they come together in combination, you have things that, like, they can receive the presence of collagen. Some of them are only used by white blood cells. Some of these combinations can detect lamin. These are all things that are found just outside of the cell, right? And these are things that are found in the extracellular matrix, right? So these are all the proteins, protein fibers, and different things that are immediately outside of the cell, right? As part of that cell being part of a tissue. Okay. So there are 24 different alpha beta combinations that when they come together in these different combinations, the actual extracellular binding domain is a peculiar shape to detect a particular product, right? And so the way they work then is by binding to the extracellular matrix, right, that causes the receptor, the integrin, to change its shape in the cytoplasmic end. And by changing its shape, it then begins to interact with different components of things like the cytoskeleton, right? So these large pink things are different types of actin fibers that make up the cytoskeleton. Right? And normally these might be kind of maybe loosely spread apart, right? But if you started bunching them together, you can imagine this would be reshaping the cytoskeleton, therefore reshaping the cell, right, to do something. How are they brought together? They're brought together when they start hooking onto these integrins that are activated. Right? So if the cell is touching a particular spot of, let's say, extracellular matrix, in that spot on its surface, you'll have a whole bunch of these integrins all bunched together, right? Because they're all attaching to this aspect of the ECM. And that means on the other side of the membrane, right, you'll have the cytoskeleton being bunched together at that spot, right? And so that controls things like cell adhesion, and it also has a big impact on the ability of the cell to potentially be mobile and move around. Okay. And then this is just an illustration. So this is a cell on a surface of plastic, tissue culture plastic that's been treated with proteins to simulate the extracellular matrix. And they've stained these different cytoskeletal fibers in them. And what's really cool is everywhere you see an orange dot, right? This is where the integrins have been brought together at that spot, because this is actually the spot where the cell is touching the extracellular matrix on the plastic. And so you can see all of those integrins then are bound on the inside of the cell to the cytoskeletal network. And that's what's going to control, let's say, the cell knowing that this is the leading edge here, right? And help the cell kind of move around or grow on this surface. All right, let's go ahead and stop there.